Good afternoon, ladies and gentlemen. I'm Eileen Han, Technical Marketing Director at CJ Bio Animal Nutrition and Health. My topic for today is gut health, and I will walk you through how we discover the components that are hugely relevant to gut health and eventually leading to optimum performance. These are the contents of my talk. Firstly, I will discuss the importance of swine gut health and how it can improve the performance. Next, I will introduce our criteria for measuring gut health. Finally, I will share the result of the optimal blend solution as evidence of our approach. Swine gut is constantly exposed to challenging environment through feed consumption. This feed is often contaminated with pathogen, mycotoxin, and endotoxin. Additionally, it contains anti-nutritional factors such as phytate, soluble NSP, and allergenic protein that stress the gut. As a consequence, the mucosal microbiota can become disrupted, and excessive inflammation can lead to damaged intestinal epithelial cells. These factors ultimately result in reduced nutrient utilization and growth, as a body energy is being consumed to deal with all these problems. It is important to note that the function of intestine is not limited to food processing and subsequent absorption of nutrient. Animal experiments and some human data have shown that intestine communicate with bacteria to support digestion through its enzymatic capacity. The gut also regulates epithelial and immune function that are important to gut health and overall health. Furthermore, the gut communicated with the brain through the vagus nerve and certain hormones, providing information about energy absorption and other conditions that could affect mood and general well-being. Finally, the intestine reduces tolerance of the immune system against its own cell while establishing an immune defense against external aggression from the food transporting the organism. The importance of this topic is well demonstrated by the increased research activity in this area. The search on PubMed using the keywords such as intestinal health and microbiota in pigs provide over 1,900 peer-reviewed papers since 1960. It is worth noting that 90% of these papers were published since 2010, indicating a surge of interest and challenges related to intestinal health and microbiota in pigs, especially during the last decade. Moving forward, I will now introduce our journey that CJ Bio Animal Nutrition and Health embarked on to find the most optimal blend of the gut health solution. To ensure our biomarkers were relevant to intestinal bile acid function protection, which was concept we used to select the best possible gut health killer items, we conducted a thorough analysis of database, scientific research, and commercial experiment. Additionally, we utilized an AI machine learning system to derive a long list of potential killer items, and this process helped us to establish our criteria for measuring gut health and select most effective gut health solution. From the long list of the potential biomarkers, we further trim it down to a short list that ensure excellent protection of intestinal bile salt function. To determine the optimal blend, we conducted the in vitro and ex vivo experiment, and the final selection was based on scientific research that include economic feasibility test. Initially, we defined 3 different categories of gut health parameter to improve the performance. To measure immune status, we selected 8 biomarkers including pro-inflammatory cytokine, to assess the level of inflammation. For oxidative stress, we measure malondialdehyde, protein carbonyl, and immunoglobulin A and immunoglobulin G for humoral immune responses. We also measure villi height and Krebs test ratio, as well as Krebs cell proliferation to evaluate effective digestion and absorption. Lastly, we analyze the mucosal microbiota to understand its relationship with gut health. AI learning machines are powerful tools for discovering items that promote gut health. By analyzing large data set of microbiome and gut health information, AI algorithms can identify patterns and correlation between specific items and gut health outcomes. We first define the target with specific biomarkers and then utilize AI-based natural language processing. The machine learning was processed from the database pool after the prediction model was established. Ultimately, we were able to drive the best possible result for the killer items. Based on AI machine learning processes, we concluded that BSHI, biosort hydrolase inhibitors, and short-chain fatty acid are the best possible candidate as killer items for gut health solution product line. For BSHIs, the finalists that we selected are catechins and vitamin K. Among short-chain fatty acid, we chose butyric acid to be included in the solution product. The first part of experiment result I would like to share with you are our in-house in vitro and ex vivo test result. Before I introduce the result, I'd like to explain to you about correlation between BSHI, butyric acid, and gut health. Conjugate biosort also have antimicrobial properties and can help maintain the balance of gut bacteria. However, if the balance of gut bacteria is disrupted, some bacteria can produce an enzyme called biosort hydrolase, which deconjugate biosort, leading to decreased antimicrobial activity and potential negative effect on the gut health. Butyric acid serves as primary energy source for the cells lining the gut and helps to maintain the integrity of the gut barrier. It also has anti-inflammatory property and can reduce inflammation in the gut, which is important for preventing and treating a variety of gut-related disorders. And it helps produce antibodies in the gut lining, as butyrate can induce TGF-beta. Our application center has established the method to evaluate BSH inhibition efficacies. Both BSH inhibitors and BSH producing bacteria, the strain, were tested in different substrate. And then inhibition levels of material were measured by absorbance. The optimal formulation ratio among the material were designed using response surface design analysis with stress induction, and efficacy of the material were more expressive, which were confirmed by ELISA and RT-qPCR. These results shown in the slide tested against E. coli at different level of conjugated biosort. As shown in the result, the responses were dose-dependent, demonstrating the relation between the conjugated biosort and suppression of pathogenic bacteria growth. Among the material tested, the several catechins were evaluated to select the best performing one. To confirm the anti-inflammatory and antioxidant efficacies, we conducted the cell culture-based in vitro experiment using the porcine intestinal epithelial cell line. we tested several ratios between catechin and butyric acid and catechin and vitamin K. Finally, the response service design method was used to determine the best optimum blend ratio among the material. The last part of my presentation is the result from our in vivo trial done in university and research farm and commercial farm. This university trial, the experiment aimed to assess the effect of CJ Gut Health solution product on the growth performance and gut health of the weaned pigs. A total 192 piglets were included in the study and monitored for 28 days after weaning. The piglets were divided into 6 groups: a negative control that received only basal diet without zinc oxide, positive control group 1 that received antibiotics, and positive control group 2 that received 2000 ppm level zinc oxide, and 3 treatment groups that received different concentration of GutLuk 0.05%, 0.1%, and 0.2%. The growth performance was evaluated based on the average daily gain, average daily feed intake, and feed conversion rate. The gut health was evaluated based on also immune indicators such as TNF-alpha, interleukin 6, interleukin 8, immune globulin A, and immune globulin G, as well as frequency of diarrhea during the study period. It was encouraging to see that all treatment groups had significantly better ADG compared to negative control group. It's also interesting to note that 0.2% CJ Gut Health product has the same ADG as zinc oxide 2000 ppm level group. Additionally, the absence of diarrhea in all treatment group and decreasing trend in the negative control group over the time suggested that CJ Gut Health product may have had a positive impact on gut health of the pigs. It is also important to note that the lower level of cytokines such as TNF-alpha and interleukin 8 indicated a decrease in inflammation in the jejunum tissues, which can be indicative of improved gut health. Therefore, these results suggest that CJ Gut Health product may have anti-inflammatory property that can benefit gut health in weaning pigs. The other immune indicator we measured in the experiment were immune globulin A and VC ratio. Immune globulin A was the indicator for mucosal humoral immunity, and 3 of CJ Gut Health product treated groups all indicated significantly higher immune globulin A level than both negative and positive controls. Villus height versus creep test ratio is another indicator used to assess the status of gut lining for proper absorption of the nutrient and maintenance of the intestinal epithelium. CJ Gut Health product increased VC ratio compared to both negative and the positive control, indicating improved gut health and immune absorption. Once we achieve positive result from the university experiment, we move on to the test in commercial environment. At one of our customer research farms, we tested CJ Gut Health product against not only basal diet but also antibiotics and organic acid-based gut health product. The parameters we measured were the ones for growth performance and the frequency of diarrhea. There was no effect of CJ GutLuk on contributing to better ADG, but however, the statistically better result than organic acid-based gut health product was achieved even in ADG. So when it comes to diarrhea, there was a high level of diarrhea in basal diet group, while all treated group could reduce the level of diarrhea. Among them, CJ GutLuk performed the second best after antibiotics group, and even at week 2, GutLuk group and antibiotics group had the same result. This trial was conducted at complete commercial farm. So here we compare only between basal diet and 0.2 GutLuk. However, all 3 performance parameters were measured and 4 different immune indicators were measured from the jejunum tissues once trial was complete. In this commercial trial, we achieved significantly better ADG in the treated group compared to negative control, although we were unable to conduct statistical analysis. It was really significant. For immune and inflammation indicator, TNF-alpha was significantly lower in the treated group, and then VC ratio was significantly higher. The treated group also showed the lower level of oxidant parameter such as marondialdehyde and then protein carbonyl. The indicator for the level of intestinal epithelial damage, Ki-67, was also lower in the treated group. As a conclusion, take-home messages. CJ Bio Animal Nutrition and Health has conducted extensive research and testing to develop GutLuk, a solution product designed to replace zinc oxide and antibiotic growth promoters in swine industry. The GutLuk has been shown in both university and commercial trials to improve growth performance, reduce diarrhea frequency, and improve gut health and immune indicator in weaned pigs. GutLuk offers science-based sustainable solution for a swine producer looking to improve the health and growth of their animal while reducing reliance on antibiotics and other growth promoters. Thank you very much for your attention, and if you have any question, you can leave the message and we will attend your inquiry as soon as possible. Thank you very much and have a good day.